Sperm cells ship out new genetic typos every month of a man's life. By the time some men are grandfathers, as many as 1 in 20 of those shipments carries a mutation serious enough to cause disease in a child. Here's the strange thing about making a baby. Men and women are running 2 completely different shipping operations. A man's body operates like a factory that never shuts off. New sperm rolling off the line every single day, from puberty until the day he dies. A woman's body is more like a sealed vault. Every egg she'll ever have was packed and stored before she was even born, decades before she might use one. By birth, that vault typically holds somewhere around 1 million eggs already packed and waiting, and none of them will ever be replaced. 2 totally different systems, both trying to deliver a clean genetic package to the next generation. And when scientists finally got precise enough tools to check the quality control on both operations, what they found breaks almost everything you'd assume about how aging damages our genes. Start with the factory. In 2025, a team at the Wellcome Sanger Institute, working with the TwinsUK study at King's College London, sequenced sperm from 81 men between the ages of 24 and 75. A modest sample size by genomic standards, though enough to establish a clear trend, using an ultra-precise DNA reading method, sensitive enough to catch a single wrong letter among millions. Published in Nature, their results showed the factory's defect rate doesn't just creep up. It climbs in an almost perfectly straight line, adding about 1.67 new mutations to a man's sperm DNA every single year, with no plateau in sight. 2 distinct mutational signatures, both already linked to normal human aging, were driving that climb. And despite the steady rise, sperm still mutates far more slowly than most tissue, roughly 8 times slower than blood cells, which is part of why scientists long assumed the sperm-making process was unusually well protected. This sequencing shows that protection has a real limit. Here's the part that should worry the quality control department. The researchers identified 40 genes where certain flawed sperm cells actually outcompete the healthy ones for space on the shipping line, a process called positive selection, where a mutation doesn't just survive, it actively spreads because it happens to give that one cell a reproductive edge over its neighbors. Most of these genes are tied to childhood developmental disorders and cancer predisposition syndromes. The result? By a man's older decades, somewhere between 3 and 5% of his sperm, potentially 1 in 20, carries a mutation capable of causing disease. And here's the detail that really undercuts the story we tell ourselves about aging. The team checked whether smoking, drinking, or body weight sped up this defect rate. None of them did. Whatever's driving this clock, it isn't lifestyle. Now the vault. Since a woman's eggs are all packed before birth, you'd expect the vault to be frozen in time. No new production, so no new defects, right? But the packing process itself is where things get complicated. When an egg finally gets used, it has to sort its chromosomes into perfectly matching pairs, splitting them evenly in a specialized division that only eggs and sperm go through. A 2022 review in Nature Review's Molecular Cell Biology, led by researchers Chloe Charalambous, Alexandra Webster, and Melina Xu, laid out just how often that sorting process fails. And the failure rate doesn't rise steadily with age. It follows a U-shape. A science study the review draws on, led by geneticist Jennifer Gruhn's team, tracked chromosome sorting in eggs from women aged 9 to 43 and found sorting errors are actually highest in the very youngest group, drop to their lowest through a woman's 20s into her early 30s, then climb again, sharply, from the mid-30s onward. 2 completely different failure modes are hiding inside that same U-shape. In teenage eggs, whole chromosomes get left behind entirely during the split. In older eggs, the molecular glue holding chromosome pairs together, proteins called cohesins, stocked once early in life and never restocked, gradually wears thin over decades in storage and lets go too early. Different mistakes, same warehouse, same end result. A mispacked shipment. So the vault clearly isn't immune to time, except for one part of it. Mitochondria, the power generators packed inside every cell, carry their own tiny separate loop of DNA inherited only from mothers. In 2025, a team led by Barbara Arbuthuber and Katerina Makova at Penn State used the same kind of ultra-precise sequencing to check whether that mitochondrial DNA picks up damage with age, the way it does almost everywhere else in the body. They compared eggs against blood and saliva from the same women, aged 20 to 42. Pulling usable DNA from a single egg cell is a technically brutal task. Each egg holds only a sliver of the genetic material found in a single drop of blood, which is part of why this kind of direct comparison hadn't been done before. In blood and saliva, mitochondrial mutations climbed with age, exactly as expected. In eggs, they didn't move at all. Flat. Practically zero, no matter how old the woman was. The team found a clue why. Mutations that would actually break a working protein were rare, while harmless background mutations were common. As if some filtering system keeps quietly deleting the harmful copies before they can spread. Biologists call this purifying selection. It's the mirror image of what's happening in sperm. Instead of favoring bad mutations, the egg vault appears to be actively screening them out. Line up these three results, and a stranger pattern emerges. It's tempting to think of genetic mutations as simple wear and tear. The longer a cell's been around, the more scratches it picks up. That's not actually what either story is about. In the sperm factory, mutations climb so steeply not because of random wear, but because certain flawed cells are winning an internal competition and getting preferentially shipped out. Which is also exactly why removing lifestyle factors like smoking or drinking doesn't slow it down. You can't outclean your way out of a rigged competition. The mutation clock is being run by biology's own internal politics, not by anything happening outside the body. In the egg vault, mitochondrial DNA isn't ageless because nothing happens to it. It's ageless because a quiet deletion process keeps erasing the mistakes before they can spread. Two vaults, two totally different outcomes, both driven by active selection, not passive decay. Aging isn't doing this, selection is. So the data actually gives two different answers, not one. For eggs, chromosome sorting errors are consistently lowest through a woman's 20s into her early 30s, then climb again from the mid-30s on. For sperm, there's no equivalent floor or ceiling. The mutation count just climbs in a straight line every year from young adulthood, no matter a man's habits. None of this is a countdown clock. It's a map of two shipping operations running on rules scientists are only now able to read. And rules that can be read can eventually be worked with. If you want more real research turned into stories like this one, stick around.